So the next point on the agenda is a discussion about how we could put together a first paper. And this is a really important activity on many levels. This is going to provide us the exposure to the outside world beyond the zebrafish community about the existence of a DANIO-CODE program. But more importantly, it will hopefully also be a demonstration of the utility of the data and demonstration of how fantastic zebrafish can be to answer biological questions. And therefore, it's very important that we make some decisions about the ambitions about this paper. And initially, when we had a chat in London, we probably already agreed that what we would like to aim for is not only a resource paper, but something beyond that, and show novel biologies with reanalysis of the existing published data sets. In other words, it show the power of a DANIO-CODE approach, and that should be sufficient and to go to one of the top journals, and for example, Nature Genetics, and announce DANIO-CODE that way. So we do have this dual ambition of a resource paper as well as showing novel biologies with it. I had a couple of conversations with two of the Nature Genetics editors who appeared to be enthusiastic and receptive towards the idea. Next year will be an anniversary of Nature Genetics, and they're planning papers along the lines of model systems and animal models. So hopefully we will be able to contribute once the reanalysis has been done. So what we need to agree on is how we're going to reanalyze data to tease out novel biologies which are already present in the published data sets, which do not interfere with individual laboratories' interests, and it's a joint effort. So, but let's go step by step. So the first is, of course, the resource. And many of these are obvious objectives, is how to generate an accessible and standardized and mineable epigenomic data set in one hub. And what we'll add to that, would like to add to that in the process, is improved visualizations, for example, of developmental dynamics, and I'll come back to that in a minute. Since many of the data sets are independent repeats by different laboratories, this potentially can be very powerful. Of course, it can represent a lot of problems of standardization, but also provides the opportunity to get more robust predictions from data, having more repeats. We will hopefully provide a database which is more comprehensive than existing databases. Well, most of them don't even exist in zebrafish, for cis regulatory elements for non-coding features. And as another resource type of objective is to integrate spatial expression data for the many thousands of genes. This is available in ZFIN with expression data coming from next generation sequencing approaches. And some of the techniques to use to integrate that may come from modern code approaches, and I guess Ben will talk about that a little bit. In regards of novel biologies, what can we aim for? I mean, there's some ideas here, and you're all welcome to contribute with ideas and suggestions or criticism what these could be. But I believe that there should be an opportunity for predictions about relationship between epigenomic events, because we have very high resolution data sets in terms of timeline, and we will be able to map when various events occur on the genome in regards of histone modifications, DNA methylation, transcription, and so on. And therefore, we should be in position to suggest what are predictive for downstream events and potentially instructive or instructive. We should be able to detect novel cis-regulatory elements, novel transcript features in the context of development and vertebrate development, perhaps in general, as long as we can generalize from zebrafish. And we will be able to classify non-coding elements further using the multi-dimensional data analysis, and I assume Ben will have a few words about that. We may be able to predict novel regulatory interactions between epigenomic features, gene expression, and gene structure. We will look for DNA codes and their relationship with epigenomic features using a developmental context. And there might be, and there should be, many other potentials here that you're all invited to suggest. How this paper could look like, there are several models, and I stole this slide from Ben, from one of his previous presentations, where he listed the two types of models of previous ENCODE activities, and what he called the ENCODE model and the mod ENCODE model. and going for little or no original science, but more towards a large resource and pointing out the biologies in independent papers, where authorship is less of an issue, as these are the words of Ben. So and there's the modern code model, where you also integrate the analysis to provide new observations from previously published data. And then, of course, authorship is a more complicated matter, but we need to discuss that and need to agree on it. We'll come back to this. Here's an example from Flybase of a gbrowse view of Drosophila data sets, and gives a very nice demonstration how you could, in an almost three-dimensional manner, represent time points of development versus tracks of data types. And this is the sort of thing which we need to think about when we're trying to visualize zebrafish data. And I refer back now to Karsten's comments on the very same issue. And the reason why this seems to be one of the interesting opportunities for developing novel visualizations and perhaps representing in a new paper is because our data is lending itself for this kind of temporal analysis across many data types. And this particular temporal analysis mostly is based on early development, because if you look at most of the published data, maybe 80%, maybe more, is covering earliest events of development when from the fertilized egg a pluripotent cell mass is forming and then starts differentiating. And this transition, the maternal to zygotic transition, is correct, as you all know, characterized by a huge changeover of the transcriptome, which was inherited from the mother in the egg, to the activation of the zygotic genome. And as this slide demonstrates, this event is generic. It happens in most animal models, including zebrafish. Therefore, what we see in zebrafish is potentially representative for many animal models, including human. So if we then zoom in a bit more to the data types, this is a slide which is to demonstrate those developmental events in terms of the actual morphology through the animation, and just symbolic ovals supposed to represent various time points in development for which a variety of data sets exist in different colors. Do not look at the position of the ovals, they're very symbolic, they're not representing the actual times. The point is to be made here is that we have lots of data points for a variety of data sets across this critical period of development. And this is perhaps where our first paper should concentrate in its reanalysis, because that's where we have most of the data, and that's where we have data which is novel in comparison to any other model system which was studied before. So it is the obvious target, perhaps, but can be debated. Now, how to deal with authorship? I really don't want to go into sort of details of this, but just want to flag it up that we invite everyone who submitted data to be part of this. But I would imagine that simply providing the raw data would not be sufficient for authorship. What we would want people to do is to contribute to this paper through helping with the reanalysis, helping with the visualizations, helping with additional data sets perhaps, and invite everyone, all those of you who already registered and those who will register in the future, to contribute to this paper. We would want to exclude anyone. What is very important, however, is that we do have some kind of groupings which coordinate activity of potentially many people. Where's the little arrow? Which way do I have to go? Oh, here we are. And would like to propose that we will need a manuscript writing group for a smaller number of people who are coordinating that activity. We need a data processing group who will coordinate that activity, data visualization group, and so on. Today is not the time to decide on this. It really is just to flag it up and ask everyone who's watching this stream or will look at it later to come forward and propose their contribution towards the paper. What is very important is that we will have to meet to discuss the details of it, and my proposition is that we meet at one of the zebrafish meetings. That's probably the easiest way of getting everyone together relatively cheaply. And there are 3 upcoming events I listed here. There might be more which I missed, which are potential candidates for getting a DANIO-CODE meeting together. Either the next one at Asilomar, the PI meeting, which will be in January next year, I think it's probably timely because by that time we will have the data sets remapped and available on Track Hub and can think about putting some paper together. Bit later, there is a smallish genomics meeting with lots of zebrafish and other groups, which would be of interest to people. It's not yet publicly advertised on the internet, but please come forward if you're interested in this meeting. There are lots of high-profile speakers from relevant fields of development of genomics, of epigenomics, and computational biology of it, which will be held in near Sevilla in Carmona in Spain in March next year. And we also will have the 10th European Zebrafish meeting, a very large conference in July. Perhaps that's rather late. We really would not need to talk before that. And there will be some limited funding available, hopefully, from the BBS-RC grant to organize this event, either attached to one of these conferences or independently. But nevertheless, put these dates in the diaries. So I think that's it from me, and can pass on now to Ben and Karsten, who will come with additional thoughts on the first paper. And of course, if there are any questions and comments, please carry on sending them in through Skype. There will be potentially some delay, but we'll be able to come back to them.
Maybe I would like to start with answering a question we got on Skype. And the question is, maybe I just read it first from Charles Vignard. About the genome annotation, do you mean gene annotation also? We probably all use Ensemble or RefSeq. Is there any plan to change, update, collaborate with these sources of gene annotation? This will be important for RNA mapping as exon junction database is used. So the answer is yes, we would like to perform gene annotation. My guess would be that for the protein coding genes, we will not find too much novel things. Maybe we find some new isoforms or so. But I think where we have the biggest potential is to find a lot of the non-coding RNAs actually. So I have in my presentation, which is already up now, I have a slide on that. But in my point of view, it also contains gene annotation, yes.
I have a comment on that. Yeah, so obviously we're discussing with EBI people and Laura Clark and Ho and some other people, both Lichak, and we will make sure that the latest version of the genome will be the reference, but additional gene annotation will be carried out, probably also with Elizabeth Bush's new RNA sequencing data and with the involvement of the EBI people. Yeah.
From my experiences with the Phantom and ENCODE, it is rather the other way around, so that as a consortium we have the new data, we provide our own annotation, and we identify genomic elements, and we provide them as a resource. And then the other, like Sanger or NCBI or whatever, they will come actually and pick up those data. And then they will use those to improve the genome annotation for the standardized models. That's what has happened in Phantom at least, and I think in ENCODE as well. It's this way. Not so much that you push them to do it, but they pull the data by themselves and then they improve the annotation based on that.